Hi my dear students, I'm Rukia Abdallah, a biology teacher. Today I'm going to discuss with you on the topic of cytology under the subtopic of organic substituents of cells. In my first presentation and second presentation, I discussed with you on a food test report writing. I discussed with you the principles underlying the report writing of food test. We also discussed the points to remember while testing food substances in a given samples. We also prepared the solution from the food. We have been given specimen A. We prepared the solution from specimen A. After preparing specimen A, we started to discuss how to go about on a report writing. We started with the starch, followed by reducing sugar, and we ended with the non-reducing sugar. Today, we continue to complete the report which we started in my last presentation. We continue with the protein. As usual, our results will be tabulated in 4 columns. Column 1 is for food substance tested. You can call it food substance tested or test 4. Column 2 is for procedure. Column 3, observations. Last column is for inference. You write here protein. Then after writing protein, it means you are required to write the procedure for testing protein. How can you go about in testing proteins? For instance, you may say that 2 centimeter cubed of solution A, where poured to clean test tube. Then what next? Followed by addition of copper 2 sulfate, 1 centimeter cubed of sodium hydroxide solution. After adding that, you are required now to add drop by drop of copper 2 sulfate solution. But don't forget the point which I shared with you in my last presentation. In that presentation, I said that in addition of copper 2 sulfate there, you are not required to add direct to the sample solution. You are required to put the drops and the wall of the container which you are using there. For example, here I'm using test tube. Therefore, the drops there should fall to the wall of test tube. Then slowly, that drop will go downwards until it reaches your solution. From there, you shake, then you observe the color change. You will continue to add drop by drop until you get the intended color. If protein is there, we expect it to get purple color. Purple coloration indicates the presence of protein. Therefore, after writing the procedure here, you write the observations, whether negative or positive. If you find that purple color was observed, you write here. Purple coloration observed in its test tube containing solution A. And if you find out that the color retained the color of reagent, which is bluish, you also say observations, bluish coloration retained in your sample solution A. From there, you can now go to the last column. You can conclude whether protein is present or not, basing on your results which you have got. For example, if you observe the purple coloration, obviously, for the case of inference, you may write that protein is present. And if you observed that the bluish color of copper 2 sulfate retained in your sample solution, then here you may say that protein is absent. Here, whether you get positive results or negative results, you are supposed to write the inference. Don't ignore. I think you have understood there. After protein here, you are required now to test the presence of lipids. I think you remember the procedure for testing lipids. You are required to take 2 centimeter cubed of solution A. You put in test tube. The test tube should be clean. Then you are required to add a few drops of Sudan 3. But in your writing there, don't say that a few drops of Sudan 3 were added. You are supposed to count numbers of what? Drops. Whether 3, 4 or whatever. When you add Sudan 3, drop-wise, make sure that you count the number of what? Of drops. For example, here you may say that 2 centimeter cubed of solution A were poured in a clean test tube, followed by addition of 5 drops of Sudan 3, and then the solution was shaken vigorously. Don't forget to shake vigorously or to shake strongly. Then the solution was left to settle for 5 minutes. Don't forget also the procedure for settling there. Then after 5 minutes, you are required to observe results in your test tube. If lipids is there, obviously you may see the upper layer stained red, indicating the presence of what? Of lipids. And if lipids is not there, you cannot get 2 layers. The whole solution will remain with the reddish color, indicating the absence of lipids. Up to here, I think you have understood the report writing. Today, I'm going to introduce to you another question. That question is this one, question 2. Why is it question 2? Because in the last presentation, I introduced to you question number 1. So I'm still continuing. Question 2 says that you have been given specimens A and B. Now we have two specimens here. You have been given with the specimen A and B. Prepare solutions from each specimen and label them as solution A and B respectively. Mind you that in the last presentation, we prepared only one solution. Today, we are going to prepare 2 solutions. The question have already been given to you. Let us check part A of question number 2. Part A says that write the procedure that you have followed to prepare solutions. Write the procedure that you have followed to prepare the solutions. Okay? Here, as we explained it previously, we are going to prepare solutions. But today we prepare 2 solutions. For instance, let us say A is groundnuts. And B, Irish potato. Let's say we have the 2 there, groundnuts and the potato. How will you go about to prepare solution from each? Mind you that for the case of groundnuts, there is no need of explaining or writing all the procedure which I gave you in the last presentation. You are not required to start with the procedure number 1, which involves peeling. Therefore, you will go directly to procedure number 2. That means you are required to cut into small pieces. After cutting, you are required to grind. After grinding, you are required to add water. After adding water, you are required to remove solid particles in order to get the clear solution. Remember that here, filtration is not needed, because when you filtrate, there is a high chance of removing all the starch. Therefore, even if the sample will contain starch, you cannot get a positive result. Also, filtration, apart from filtration, decantation also is not required. When you decant, it means, first of all, you allow your solution to settle down. Then you decant slowly. This one also may eliminate out starch. Therefore, in order to avoid that, in stage number 4 here, I just said that remove solid particles in order to get a clear solution. After getting your clear solution, then you can use it in the second part of the question, that is question 2, part B. Apart from groundnuts, you are also required to prepare solution from Irish potato. But for the case of Irish potato, you are required to start with the peeling. Therefore, you peel your specimen. After peeling, you cut or you slice potato into small pieces in order to be easy for you to grind. Then you go to procedure number 3, that is grinding. After grinding, you are required to add water. After adding water, in this part we will go up to number 5. Number 5, you are required to remove solid particles in order to get the clear solution. Then, after doing that, you are required to write your report here in preparing solution by following the principles which I gave you in the last presentation. That means don't forget to use past tense. Use past tense in preparing your solution in both cases. I think you have understood. And after getting the solution, don't forget to label your solutions. Solution which have been prepared from groundnuts, you should label solution A. Solution which has been prepared from Irish potato, you should label it what? Solution B. Don't mix the labels. That is why you are supposed to label your solutions. Then, after labeling your solutions, it means you are through with the question number 2A. Requires you to prepare solutions. After preparing, don't forget to write the procedure. As usual, you use simple past tense in writing your procedure there, starting from procedure 1 to the last procedure. Then, let us go to part B of the question. Part B says that use the given reagents to identify the substances present in each solution. Therefore, you are required to use only the reagents which have been provided to you. Don't demand for the reagents which you are not provided to you. For example, sometimes you may find that one reagent is missing there. Therefore, if you observe that, it means you are not required to test that food substance which is using that reagent. That is why it is not provided to you. Sometimes you may be asked to test only carbohydrates, for example. Let's say the question demands you to test only carbohydrates. For that case, you know the carbohydrates which you are required to test because you have covered in theory part of cytology. For example, you know that there are 3 classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. But in a food test, you are going to test reducing sugars, non-reducing sugar, and starch. Clear? Make sure that you read the question careful. You understand the question. Then you follow the instructions. Okay, let's say that you are familiar with the reagents which have been provided to you. Now you are going to perform now an experiment to identify food substances present in your prepared solutions. Solution A and solution B. How to go about? How to go about? Here, the procedures are the same. You are going to start with the starch. You take a test tube. You take a test tube there. Then, after taking test tube, you take 2 centimeter cubed of sample solution. Sample solution A. 2 centimeter cubed of sample solution A. You pour in a test tube. The test tube should be clean. After pouring your solution A in a test tube A, let's say, then you are required to add iodine solution. Iodine solution you add there in terms of drops. You add the first drop. If you find that the results were not seen clearly, you add the second drop. Then obviously you will get the blue-black coloration if starch is there. Don't use too much iodine solution. Starch is very, very responsive. to iodine. So even though when you add 1 or 2 drops, if starch is there, you can get the positive results. Clear? After completing testing starch in a solution A, you go to test for starch in solution B. But in order to avoid the mixing of things, you should write rough work there. That is A, you write A there, test for starch. The procedure, you have already written. Observation, if you get blue-black, you indicate there. Blue-black coloration was observed in a sample in test tube containing solution A. Then you go to B. You do the same. You take a clean test tube. You put 2 centimeter cubed of solution B. You add drops of iodine solution, but drops should be countable. Count and write the proper number there. Check and then observe the results. If you find out that the result there is blue-black, you say blue-black coloration was observed. But if that color is not there, you write the color which you see in the test tube there. If it retains the color of iodine, you should be specific. Which color of iodine? I think in the first presentation, based in report writing, I said that you are required to know the color of reagents. For the case of starch here, I said that iodine is yellow when it is in water. Some say that it is orange when it is in water. And others says it is brown when it is in water. Therefore, you should be very, very specific. You should be very, very specific. From what I know, the color of iodine when it is in water, it is brownish in color. But it depends on concentration. When you add more water, the brownish color changed and it looks like a brownish yellow. Brownish yellow. Or brownish orange. Therefore, concentration matters. When it is high concentrated, it appeared in a brownish color. But if concentration is less, it means when water molecules are so high, then you will get brownish yellow. I think you have understood there. Or brownish orange. From there, after writing your report concerning with the starch, you are required to go to another food substance. As I have said in my last presentation, you are required to test reducing sugar first before testing non-reducing sugar. The procedure are the same as we did previously. You take your clean beaker there, I mean test tube. You are required to use 2 test tubes. You label them, test tube A, test tube B. Then you measure 2 centimeter cubed of sample solution A and you pour in test tube A. You measure 2 centimeter cubed of solution B and you pour in test tube B. Then you add equal volume of Benedict solution in each test tube. It means you measure 2 centimeter cubed of Benedict solution and you pour in a test tube containing solution A. You measure again 2 centimeter cubed of Benedict solution, you pour in a test tube B. After doing that, we know that heat should be applied there. You are required to boil your sample solutions in each test tube. You remember the hints or the point to remember while boiling there. Don't boil directly throughout. You boil a little bit there, then you remove your test tube. You observe the changes there. You boil again for a little bit, you remove it and you observe the color change. The aim of doing that is just to get the series of color changes. We expect to get the color changes from bluish color because you added what? Benedict solution is bluish in color. So the color should start from here to greenish color, from green to yellow, from yellow to orange, from orange to brick ready precipitation. But here, last time I said that in order to reach to brick red coloration or orange, it depends on the concentration of sugar in your food sample. If concentration of sugar is little, you cannot get a brick red coloration. You may end either to yellow or orange. But whatever the case, if you get yellow, it means reducing sugar is there. You will conclude the presence of reducing sugar. If you get orange, you will also say that reducing sugar is there. If you see brick red, you also say that reducing sugar is there. Don't forget to use what? Past tense here. For example, in observation, you may say that a series of color change was observed starting from bluish color to greenish, to yellow, to orange, up to brick red coloration. Inference, reducing sugar is present. I think you remember why are we using the present tense there. I said if it is groundnuts and you find out that there is reducing sugar, for example, even if another scientist will come there after 20 years, still that scientist will get the same results. That is why there is no need of using simple past tense, but it is better to use simple present tense. Clear? Then, after getting the results, which is positive, for the case of reducing sugar, you are not required to pour out the solution. Don't throw away the solutions of your results. You are required to put it aside. Let's say you put in your test tube rack. Then you go for testing what? Non-reducing sugars. In non-reducing sugar, you're also supposed to have 2 test tubes. You should label them, test tube A and test tube B. After labeling, you measure your sample solution, 2 mils, 2 centimeter cubed of solution A, you pour in test tube A. You measure 2 centimeter cubed of solution B, you pour in test tube B. Then you measure another reagent. You measure dilute hydrochloric acid, 1 centimeter cubed. 1 centimeter cubed of dilute hydrochloric acid, you pour in a test tube containing solution A. You measure again 1 centimeter cubed of hydrochloric acid, you pour in a test tube containing solution B. Then you take both test tubes and you boil. You heat there. Why are you heating there? In order to make the reaction to go faster. I think you remember the aim of adding HCl. The aim of adding hydrochloric acid is in order to facilitate what? Hydrolysis. You are hydrolyzing non-reducing sugar to reducing sugar. After that, don't forget to remove the acid there by adding sodium hydroxide. The ratio, if you remember there, I said the ratio should be 1 to 1. If you use the 1 centimeter cubed of HCl, you are also required to add 1 centimeter cubed of sodium hydroxide. But here, if you reverse, if the sodium hydroxide is less than HCl, obviously you cannot get the results. So the amount there should be the same as have been illustrated in our books, or if it exceeds, the one which should be exceeded is sodium hydroxide and not the acid. After adding sodium hydroxide, you add again equal volume of Benedict solution. Last time I said which equal amount are we adding all the reagents which have been added there? Because we added HCl 1 centimeter cubed, we added sodium hydroxide 1 centimeter cubed. So total 2. And the sample solution is 2. When you add 2 plus 2, you get 4. Then add equal volume of Benedict solution. Which volume are you going to use? You are going to use the volume of your sample solution. Because Benedict solution there, go there and it reacts with your sample solution. It means you will add what? 2 centimeter cubed of Benedict solution in each test tube. After adding there, as usual, you are required to boil. The style of boiling is the same. Boil for a while, remove your test tube, observe the color change, boil again for a while, remove it, observe the color change, continue to do so until you get the expected results. After getting the results for non-reducing sugar, you are now required to compare with the results in reducing sugar. Let's say this is a test tube containing the results of reducing, and this one is the test tube containing the results of non-reducing sugar. You compare the two. You compare color intensity. If you find that here you ended with the yellowish color, and in this one you also ended with the yellowish color, it means reducing sugar is present, but non-reducing sugar is not present. Clear? Do you know why? For instance, the amount of reducing sugar, let's say, is 1 gram in your test tube. It is just an example there. And here, let's say you have got yellow color in A. Test for reducing, let's say. in the same solution, you tested non-reducing, still you obtained yellow color. It means that yellow color is the color of what? That yellow color which you have been obtained is due to this amount of sugar, 1 gram of glucose, for example. If, let's say, sucrose is there, it means when hydrolysis takes place, obviously, sucrose will provide another amount of sugar. Therefore, concentration of sugar will be high compared to test for reducing. So, in order to indicate the presence of non-reducing, the color intensity should go beyond what? Yellow. For that case, should go to beyond yellow because of increasing of what? Concentration. Clear? But sometimes you may test reducing sugar and you will end up with the brick-ready correlation. And you will test non-reducing sugar, you will also end up with the brick-ready correlation. There, the criterion of color intensity cannot be used. You are required to use another criterion. And that criterion is what? Amount of precipitation there. Amount of precipitate. If you observe the amount of precipitate is the same, it means there is only reducing sugar and no non-reducing sugar. But if amount of precipitation is more in a test for non-reducing sugar than reducing sugar, it means reducing sugar is present, also non-reducing sugar is present. Clear? Make sure that you practice in order to be familiar. Okay, after testing non-reducing sugar here, you are required to go to another food substance. That is, you go to proteins. You remember, you know the procedure for testing proteins. You take your test tubes which have been labeled A and another one B. You measure 2 centimeter cubed of each and you pour to the respective test tubes, followed by addition of sodium hydroxide there. You remember the amount. Then you put drops of what? Drops of copper 2 sulfate. You put it drop wise. You remember, you are not required to put it direct to your sample solution. Put in there. The drop should fall to the wall of your test tube. Then it will go slowly up to your solution. Then when you shake, you will see the results. When you observe the purple color, you will conclude the presence of proteins. Clear? But don't forget to use what? Don't forget to use past tense for the case of procedure and observation. But inference, you use simple present tense. Clear? After that, you are required to test lipids in both cases. You measure your sample solutions, you pour in each respective test tubes, you add a few drops of Sudan 3 in each case, but I said don't write a few drops, count the number of drops there, then shake it strongly. After shaking strongly, put the solutions aside. Allow them to settle for, let's say, 5 minutes. After that time, check your results. If lipids are there, we expect it to get the upper layer, which is reddish in color. They are found on top of water and they will stain reddish in color. The reason you know, because we have already discussed the basis of each test there. After that, you write your report. You write your report. Don't exclude the process of shaking vigorously. Don't forget the stage of putting the sample solutions aside in order to settle for a given specified time. After that, after finishing writing your report, then you are required to go to another aspect. But here in report writing, when you have been given 2 or more solutions, some students construct so many tables. They construct table 1 for solution A. They construct table B for solution B. They construct table 3 for solution C, let's say. They construct table 4 for solution D, for example. This is not proper. It is time-consuming process. You will use a lot of time repeating the same procedures, the same what? The first and the second column will be the same. Why are you doing several times? In order to minimize time, construct only one table. Even if you are given so many solutions, use only one table. For example here, for the case of our question with the 2 solutions, you can say that test for starch. 2 centimeter cubed of solution A and the solution B were kept in their respective test tubes, followed by addition of 2 drops of iodine solution in each case. You are through with what? Procedure. Observation. Blue-black coloration was observed in a test tube containing solution A, for example, and solution B retained the brownish color of iodine solution. Inference. Starch is present in A. Starch is absent in B. You see? So even if you have been provided with so many numbers of solutions, construct only one table. It saves your time. It also saves what? Papers. You cannot use many papers. Clear? And after the report, you may be provided with the other questions. For example, you may be asked, which food substances have been identified in solution A and B? You are required to write it. What is the sources? What is the sources of food substance identified in A? You are required to write it. What are the sources of food substances identified in A and B? What is the main role played by the food substance identified in B in living organisms. Therefore, you should be familiar with those kind of questions. You should know the natural source of starch, reducing sugars, non-reducing sugars, proteins, fat and oils. You should know that. You should know the functions of starch, reducing sugar, non-reducing sugar, protein, lipids. You see? You should know any products of digestion of the food substances which have been identified there. You should also know the enzymes played a part in conversion of that food substance into another form. You should be very, very familiar to that. Clear? Therefore, up to here, I have reached the end of my presentation. Don't forget to read materials concerning with the report writing of food test. The question is so simple. Therefore, don't complicate it. Make sure that you make things to be simple in order to get the intended objectives. Clear? Use your books. Use Thai book, Form 6 Biology Student book, which has been written by Thai. Consult your BS, 4th edition. Use also Understanding Biology, 4th edition. You can get the materials of food test from there. Clear? Okay. Thank you. We shall meet next time.